This protocol offers an easy and rapid approach to screen candidate biomaterial inks for potential cytotoxic effects against sensitive primary and immune cells prior to complex 3D bioprinting experiments. After incubation of the immune cells on the biomaterial ink constructs, the cells can be easily retrieved from the constructs by gentle washing to perform downstream cytotoxicity or biomarker analyses.
This protocol is directly relevant to tissue engineering, in which bioprinted tissues that are destined for transplantation must be constructed from primary cells and biocompatible biomaterial inks.
To begin, prepare a BioInk cartridge for installation onto the Incredible Plus 3D bioprinter. First, remove the blue end caps from the 3 ml biomaterial ink cartridge and affix a sterile 22-gauge conical bioprinting nozzle to the luer lock end of the BioInk cartridge. Connect the air pressure supply tubing for printhead 1, or PH1, to the opposite end of the cartridge, and insert the cartridge with its attached conical bioprinting nozzle into the vertical slot of PH1. Next, firmly seat the cartridge in PH1 with a conical bioprinting nozzle extending below the printhead. Tighten the screw on PH1 clockwise until finger tight to lock the BioInk cartridge in place. Note that the 3D bioprinting can be performed with PH2 left empty. Power on the bioprinter and launch the bioprinter software on a PC connected to the 3D bioprinter via a USB 2.0 cable. In the software, click the Connect button to sync it with the 3D bioprinter. Observe